Thank you, and thanks for the invitation. So I come from the governmental research organization VTT in Finland, and we do a lot of work in the biorefinery-related aspects from biomass degradation, enzyme production, to metabolic engineering and chemicals production. But I thought that I today talk about bioethanol production because it actually gives a very nice example, in a way, over the time, what problems people have been facing in trying to produce bioethanol from biomass sugars by yeast. And we are working with Baker's yeast Saccharomyces cerevisiae, and that's probably obvious for most of you why it's a very good organism. Of course, there is also excellent genetic and molecular biology tools, a very vast amount of physiological data. It was the first eukaryote with sequence genome, and this, of course, has also led that it's a very good organism for systems biology studies, and there was also a genome-based metabolic model constructed 2003 for this organism. But if we think about synthetic biology or metabolic engineering, so there are some challenges in comparison to bacteria. For instance, it's really because of being a eukaryote, you have compartmentalization of the reactions. So for instance, we do not know very much how the different metabolites are transported to and out from mitochondria, and also nucleus makes complications in the regulatory networks, and there are also even indications that some of the glycolytic enzymes would have a role in the nucleus, which we do not understand yet too much. There are genes of unknown functions, still about 30% of the genome, and what is also particular for Saccharomyces is that there has been a genome duplication, so part of the genes are duplicated, so there are redundant genes, and of course this means that you could have backup reactions in the cells and also many unspecific site reactions. And then when we talk about real production, so we would like to use the industrial strains. This is giving sound. And then many of these are polyploid, aneuploid, and of course then you have the allelic effects, and if you want to do mutations, you may have difficulties in finding what you want or the effects of the mutations. And obviously there is still quite a lot of lack of understanding of robustness, real industrial robustness of the strains. So, but it is a very favored organism because it's generally regarded as safe. It's easy and cheap to cultivate, which is important. It can grow aerobically and anaerobically. And what is even more important is that it really is the organism which can be used in production in very large scale, like in ethanol production. It's excellent in ethanol production, and the tolerance is very good. Also, the pH tolerance of the strain is good. It's not prone to contaminations, easy to separate from the medium. There are lots of industrially robust strains, and what is also important is that the strains, some of the strains are really tolerant towards the inhibitors, which you find when you hydrolysate lignocellulosic materials. And there are various examples of development and metabolic engineering of Saccharomyces for production of various chemicals, including bioethanol, other biofuels, lactic acids, specialty chemicals. And we are interested in many of these. For instance, we have been working quite a lot with various yeast on lactic acid production, but as said, I'm talking about bioethanol now, and obviously it's a very bulk product, so what you need is to have close to theoretical yields and very high production rates of the product. And what we want then is to use these waste plant materials and the sugars which are derived from these materials for metabolic engineering and conversion then to bioethanol. So Saccharomyces is good in using monomeric sugars and and some of the disaccharides and trisaccharides takes them efficiently into the metabolism, and even some disaccharides here, because they can be split then into the monomeric sugars, and this is because you can the yeast can secrete itself enzymes or be made to secrete enzymes to the medium, which can then hydrolyze the sugars. And you can even think about producing cellulases from other fungi, filamentous fungi, in yeast, so that some of the cellulose could be converted to cellobiose and glucose, and that is how you can then get even cellulose used by the yeast. Also, I would say that almost all hemicellulases originating from other fungi can be secreted from the yeast to the medium. But what then is a problem is that the sugars which are released from the hemicelluloses, like xylose, arabinose, and galacturonic acid, which is coming from pectin, for instance, rich in sugar beet pulp. So there is blocking metabolism of these sugars within the yeast cell. And this is what you then need to start engineering. And if you look at then the different relevant biomass sources and see how much actually they contain these hemicellulases, the percentage here, you see that it is high. So you really need to be able to use the other sugars as well, the pentose sugars. And just to show you now the central carbon metabolism of Saccharomyces, so that you get used to my pictures. So here is here is glucose converted through glycolysis to ethanol. And what we have now then done over the years is to identify pathways for these alternative utilization of these alternative sugars. First of all, xylose here. So a fungal pathway from other fungi, you can have a pathway for xylose utilization where you have the xylose reductase and xylitol dehydrogenase, and then dexylulose formed, which then feeds into the pentose phosphate pathway, and then hopefully to the glycolysis. We have also characterized the fungal arabinose utilization pathway, taking genes from yeast and filamentous fungi, and then we have also characterized the pathway for galacturonic acid utilization, which feeds into the metabolism here. But I will now talk about more on the xylose utilization pathway, because here really we have analyzed, and also the other groups which are mentioned here, lots of the limitations in xylose fermentation. So the thing is now that we would then need to shuttle xylose here through the xylose pathway to the pentose phosphate pathway, and then get it into the glycolysis and to ethanol. And the limitations there, first of all, is sugar uptake. Because when the genome of Saccharomyces became available, so it was noticed there are lots of permeases for sugars here, and these are hexose permeases. But none of these actually are specific for xylose. They can transport xylose, but the Km for xylose is very much higher than for glucose. And people, of course, tried, us included, to fish out different permeases, xylose permeases, from other fungi or from bacteria. But none of these efforts have so far really given superior permeases which would transport xylose. And thus, then, there is still a problem of co-utilization of glucose and xylose, which you find in the lignocellulosic hydrolysates. Another bottleneck is the pentose phosphate pathway reactions, and it became quite early clear that you have to overexpress the xylokinase here, and this by overexpressing that you get then anaerobic fermentation of xylose in minimal medium. But you also have a positive effect if you overexpress the pentose phosphate pathway reactions, transaldolase and transketolase. So this shows that it's important for ethanol formation from xylose to increase the flux through the pentose phosphate pathway. Furthermore, then one could think that, okay, are there general metabolic regulation limitations, whether xylose is able really to shuttle into the glycolysis, activate the glycolysis as a sugar the yeast has not seen before, whether there are respiratory requirements in xylose utilization, and how then actually, if you feed in xylose and not glucose, how is the whole regulation of the ethanol formation pathway and the glycolysis? For instance, glucose 6-phosphate has been thought to play a key role in sort of regulation of this pathway, and now in principle we don't have it. So the question also is whether we can really ferment xylose or whether respiratory metabolism is needed. And to analyze a little bit of this, we did quite a lot of genome-wide analysis, transcriptional profiling, and proteomics experiments where we studied xylose versus glucose in anaerobic, aerobic conditions, in batch cultures, chemostat cultures. And I don't go into detail into the result, but just to show that there are certain reactions which are always upregulated on xylose versus the other conditions, which is the upper part of the pentose phosphate pathway here, leaning here to the glycolysis, and then the pathway to acetate production, and also pathway to cytosolic acetyl-CoA production and some ergosterol genes. And what also has come up from these studies is that it seems to be that Saccharomyces is sort of confused whether it's fermenting or respiring when it uses xylose, so that for instance, the TCA cycle, respiration, glycosylate cycle, and gluconeogenesis enzymes are not so repressed as what you have on glucose. So xylose clearly then is not a fully repressing carbon source. And what was also important or interesting to notice was that some of the glycolytic enzyme genes actually showed different types of phosphorylation on xylose than on glucose, which indicates that there is regulatory mechanisms which we are not actually aware of yet in basic metabolism. So much of these things were then thought that could be related to the redox problems which are caused in the xylose pathway, the redox cofactor imbalance, and that is depicted here because what was seen in the early days was that actually xylitol was excreted, so actually the reaction to ethanol was already blocked here in the first phase. And this could be due to the different cofactor specificities of the two enzymes in the xylose pathway. So you can see it more clearly here. So the enzyme we are using from another yeast, Pichia, utilizes in the first reaction NADPH, it prefers that, but the second reaction is utilizing NAD. So actually what is generated here, NADP cannot be used in the second reaction, and this gives a sort of a problem for the redox balance, overall redox balance of the cell. And what you would actually want would be then that if the first reaction would be NADPH dependent, the second one would utilize NADP, so that you could could use the cofactor generated here in the second reaction, or then that the first reaction would use NADH for the second reaction to work efficiently. But people have tried, and we have also analyzed various sources for these two reactions to find appropriate cofactor specificities, but we have been not able to find those in nature. And this then, the cofactor imbalance leads to the fact, which is also supported by metabolic flux analysis, that you actually, instead of the pentose phosphate intermediate going down in the glycolysis, they actually shuffle to the oxidative part of the pentose phosphate pathway, because NADPH is generated here for the first reaction here to work. And then also that there could be really requirement for respiration to oxidize the NADH formed, in particular if one would try to grow it in anaerobic conditions, so you would need to have these reactions. And also there are other reactions in the cell which generate NADPH, like the acetate pathway here. So obviously there was lots done to try to improve cofactor specificity, for instance, of the first the XR and XDH reactions in the xylose pathway, and there is some success with both approaches. People have been, or we in particular have been searching through genome analysis and screening approaches, new pathway enzymes from fungi which would have different specificities, and we actually were successful for finding one for arabinose utilization, but not for xylose. Bacterial transhydrogenases were expressed in yeast, which could do the conversion of the cofactors, but that had no desired effect. And then, in particular, we have been trying to build up transhydrogenase cycles with different enzymes, so which could balance the cofactors. And you can see the principle here. So what we would like to do with an external enzyme here is to convert the cofactors generated in the xylose pathway to the ones needed in the pathway. And one of such enzymes we found from Chiromyces lactis, which was actually NADP-dependent glyceraldehyde phosphate dehydrogenase, which was not known to exist before in eukaryotic organisms, and it converts the reaction from glyceraldehyde phosphate to then further down in the glycolysis. And when you look then how one could use this enzyme in cofactor balancing, you can see that if one would start from 3 xylose molecules, produce 3 xylulose, which would then be shuttled into the glycolysis, producing 5 glyceraldehyde phosphate and then 5 ethanol. So if you would overexpress this new enzyme and think that 3 gap molecules would be converted by the new enzyme, so you would generate 3 NADPH molecules, which then would balance the cofactors in the first reaction here. And then there would be 2 left for the natural NAD-specific CAPDH, and this would provide 2 NADHs, and 3 NADHs would be generated here, which would then be converted by the last reaction in the ethanol formation pathway to the 5 NAD needed in these reactions there. And when we overexpress this or express this enzyme in Saccharomyces, so we saw a clear reduction in xylitol and improved ethanol formation, which showed that actually this kind of a cofactor balancing would be functional in yeast. Other things which you can do is really mutagenesis and selection for improved anaerobic fermentation. And you can see here that even polyploid strains can be improved for anaerobic ethanol production just by mutagenesis and screening. And the last thing here is the a xylose isomerase, which some of you may have been thinking, why not to mention that? And this is an enzyme which actually converts directly xylose to xylulose, so you would skip the two redox reactions here. But obviously this was tried in very early on, so that there were a number of trials with bacterial and plant xylose isomerases expressed in yeast, but there was no activity actually found. And then a few years back now, the group from Delft, Jack Bronk and Hans van Dijken, they took an XI from Pyromyces, an anaerobic fungus based on the genome, which came then available. And this gave some activity which then allowed people to go forward in metabolic engineering using that gene, so that you can then build in the xylose pathway using a single step here. However, this is not sufficient because the activity is low, so you really need overexpression of the enzyme. You also need to knock out some unwanted side reactions from the cell, and as showed previously, you need to overexpress most of the pentose phosphate pathway reactions to get efficient xylose fermentation. And furthermore, what was done was just sort of a zero-level metabolic engineering or traditional mutagenesis through evolutionary engineering, so having chemostat cultures where you selected for anaerobic growth on xylose and then verified that this also led to improved xylose fermentation. And obviously in these mutants we have still a lot of things which are not known, which made the xylose fermentation improved. As a conclusion, I could say that ethanol can be now produced with Saccharomyces with good yields from xylose. I don't give any figures because I know the published figures are not the whole truth. There is still need for improvement of anaerobic production rates in xylose fermentation, and obviously there is still much we can do to combine the individual observations which have now been sort of gathered over the years to improve xylose fermentation. Co-fermentation of glucose and xylose is needed, and possibly also arabinose. But the work shows that it's sort of a good opening, because now that you can utilize xylose as a raw material, this opens up the utilization of biomass and second-generation biomass, lignocellulosics, for production of other chemicals as well. And we have been working a lot with lactic acid, and we have also shown that that can be produced from xylose, and you can then think of other products as well. And you can then think that, or simply thinking, you just put in the lactate dehydrogenase and produce lactic acid. And you can really do that well with different kinds of yeast species at low pH, which is important for lactic acid production. Some of my last points are that I don't think my talk was on synthetic biology, but still, if we think about real life and getting production strains really for bulk products, so it is not a simple plug-in approach usually, because as you hopefully have seen from my presentations, there are various cellular consequences of metabolic engineering. One of them is obviously the redox, and that's important for almost all metabolic engineering, because they involve redox reactions. Energetics, obviously, because large-scale bulk production requires anaerobic conditions, so energetics becomes really a sort of a challenge. And export of products like lactic acid, for instance, overcoming the stress reactions in the big fermenters. And the robustness of the host, which I want to emphasize, because for real life, so you need to have various different tolerances of the strain, and even something which is in the real big fermenters, like robustness against high pressure, which you have in the big vessels. So that is why there is still a reason to actually select your host strain based on these properties. So, and some of the things which I would like you to consider also that in metabolic engineering and production of chemicals, you would need to take all the sort of tools sort of in use, like the mutagenesis and screening really gives amazingly good results very often, and in particular when you don't understand what is behind the good properties and what you would need to do with rational metabolic engineering. Also, the model-based approaches are not always giving good results for the simple reason that we don't know enough of the biology. And then what I would also like you to consider that there is actually very interesting other microbes out, and in the yeast field, for instance, many non-Saccharomyces yeasts which are listed here, which have very beneficial physiological properties for production of certain chemicals, and which also are robust, so which could be used in real life production. So thank you.
Are there any questions for Mirja? What do you think is the key pressing problem in large-scale fermentations that could be addressed with tools in synthetic biology? If there's one thing that could be fixed.
Yeah, I guess one of the things which was also related to the previous speaker or mentioned was to analyze, I mean, how much you need for growth and maintenance versus then product formation. I think that is something which you can, whether it's synthetic biology, but at least it would be understanding through the mathematical, the mathematical means and metabolic models. So I guess that's something where you really then optimize, because you have to have sort of some growth occurring or at least maintenance energy to be formed. So I guess that's something which you could be, which would be quite key.
There was famous laboratory in Finland by Virtanen a long time, and then yeast, mainly yeast was carrying in Copenhagen Vinge. I wonder if there is any relationship of your lab to this two old labs, or that traditions are lost.
You probably, the traditions are lost, I guess, already, because that was. But obviously, I mean, there is a long history in yeast research in Finland. So obviously, I mean, it still is some ways the basis.
Thank you.